It's that time of the semester where we need to start thinking about our unknown project. So I wanted to make this video just to make sure we're all on the same page of what that's going to involve. The more prepared you are for it, the better. Now, the good news is this does not involve a 10-page paper. It's mostly going to be lab work with a little preparation ahead of time, and that will be completed before Thanksgiving. And then just your little report to me of what you did is going to be due right after Thanksgiving. So let me be clear on what that will involve. Your unknown is going to be worth 100 points, so it's as much as an exam grade. Like I've told some of you, it's usually a good grade for people. So as long as you're just kind of being as careful as you can, conscientious, I guess, dotting all your I's and crossing all your T's, there's no reason that you can't get 100 on this. So what it does involve, you need to complete a gram stain on your own, and should be the correct colors. So basically, let me back up a minute. I'm going to hand you a broth test tube that will have 1 gram positive organism and 1 gram negative organism in there. So a mixed broth. Okay, and so on the first day that we are doing our unknowns, which is the same day that we're going to review the results from lab 16 and 17, you're going to want to do a gram stain from that mixed broth. Okay, and as long as you can find that on that day and show me, you'll get your full 10 points, assuming that the gram positive, right, is the right color, purple, and the gram negative is red. Okay, I always have a few students that have trouble finding the gram stain on that day, and I will allow you to do a gram stain on the next lab. You get docked a few points, but this is not an all or none thing. Okay, more about that in a second. You will need to complete some flowcharts. That will be worth 20 points total. We'll talk about how to do that in a minute. I'm going to be checking your technique on some streak plates. Okay, that's the second thing. So you're going to do this on the first day, and you're also going to do this, okay, 1 for first day. You're going to be streaking out plates because you can't try to figure out which 2 organisms are in here when they are in a mixed broth, right? You have to separate them out. You have to get a pure culture before you're trying to identify something. All right, so when you streak them on selective media, you'll get your gram positive on one plate and you'll get your gram negative on another. Okay, so very important, choosing wisely which media is going to allow you to do that. All right, the actual identification is, 50% here. It's not an all or none thing. So typically, we'll talk about that's how you can lose points on that, but typically you've done enough right to earn a lot of those points, even if you don't identify your organism correctly. And then there's just a few things that I could dock you for here and there. All right, so again, all adds up to 100 points. All right, a little bit more about that gram stain that you're going to do on the first day. Kind of lost my bullet points off to the side here. Like I mentioned, if everything kind of looks purpley red, then could get docked some points for that. Usually, though, if you just keep searching for some field, you'll find some distinctly purple cells and some distinctly pink cells. Taking care of the microscope, I'll be checking that. We haven't emphasized that as much this semester because we've left those scopes sitting on the bench the whole semester. We don't typically do that, but you should still understand that you're going to return it to low power when you're done, and then you're going to clean the oil off, throw away your slide, all that stuff. And like I said before, if you have trouble finding your gram stain on the first day, then you can definitely do it our second lab period. Just get docked a few points there for that. It's usually a little bit easier when you take your cells from a plate, but hopefully you can all do that from a broth. Okay, like I mentioned, you are going to want to construct some flowcharts, and we are going to do the gram-positive flowcharts together in lab, so hopefully you'll get a feel for how that's going to go. But you will need to construct your gram-negative flowchart. Okay, the gram-positive organisms, there are 8 of them that are going to be your possible unknowns. So there are 3 staphs. These are the ones that we began working with in lab 13, and there are 5 streptococcus. Okay, those are the ones in lab 14 and 15. Now, obviously, there's lots of other gram-positive organisms out there, but your job is to figure out, if you have one of these 8, how to, again, identify which one that you have. The 10 gram-negative organisms are the ones that are listed in lab 16 and 17. So there's lots of different genera there, Enterobacter, Proteus, Klebsiella, E. coli. Okay. You have to have all 10. Now, usually students do a gram-positive chart separate from a gram-negative chart, just because it's hard to fit it all on one page. But all those organisms need to be there. This is not an exercise where you identify your organism and then tell me the steps graphically how you got there. That's not what the flowchart is designed to be. In fact, let me show you an example of a flowchart before I go any further. This is from your textbook in chapter 20. This is an example of a flowchart to identify some gram-negative organism. And here they just have it identified by genera here. So you will have a genus and a species, not just a genera. But you're going to pick some test to start out with. And here they've used lactose. Okay, that might be a reasonable thing for you to do as well. It's nice to pick out some tests where, you've got about half that are positive and about half that are negative, but that's not absolutely necessary. Pick some tests that you feel really confident about reading the results accurately. Here, they've chosen to do motility next. I would say definitely don't choose that one, because the test that we used for motility, I'll talk about how difficult it is to read, and it's really hard to tell whether there's growth there and where it is. So I would not recommend that. But again, this is just an example. You're going to do a series of tests until you get down, with one organism at the end of each pathway. Okay, if you get down here and you say Salmonella or Proteus, you need another test to tell those apart. Okay, so one organism at the end of each pathway. Your flowchart doesn't have to have little pictures like this on it, but sometimes students choose to say, positive on this side, blue, negative on this side, green color change. Just, that's fine. I don't need to see that, but if you think it might keep you from getting mixed up, that's fine. And usually you have all the positives to the left and the negatives to the right. So you can see this consistent throughout the flow chart here. You always do the positives on the left or the negatives on the right, or the other way around. Okay, so anyway, there's an example. Like I said, you're going to have, say you're doing a gram-negative, all 10 organisms will be on there. If you miss one, you're automatically going to get docked 2 points. So count them, make sure they're all there. Make sure they are written grammatically correct. All right, so remember, the genus should be underlined, the species should be underlined, uppercase genus, lowercase species. If you're handwriting it, if you're typing this out, it should be italicized. And it doesn't really matter to me whether you type this out or whether you handwrite it. As long as it's legible, it's fine with me either way. Do make sure you spell everything right, right? This is your best work. It's right there in the book. double check it or you're going to get docked some points. You could miss points if you make a mistake in keying it out. So, for example, if you get mixed up as to what organism is sensitive to that antibiotic disc and what organism is resistant, then again, you could get docked a point there. So, if you just, tell me a result that is not true, or you get a little bit mixed up, you get docked a little bit there. And I'd ask that you don't abbreviate the genus, unless you have it somewhere, like maybe you have it at the top of your chart, Staphylococcus, and then after that, S, I'll know means Staphylococcus. But if you've got it one place, again, that's fine, but don't just automatically abbreviate all your genera when you haven't written it out somewhere. Okay, I already talked about that. All right, so don't be afraid to ask me questions about the flowcharts. I can eyeball it. Oh, yep, that's the right idea. What a flowchart is not is a table with all your results here. Okay, that's a table. Okay, I can look at that somewhere else. This is not going to tell you what test to do first, and then what test to do after that, and what test to do after that. Okay, so arrange it where you're doing one test at a time, and arrange it so you're doing one test at a time, just like this. Okay. All right. Your streak plates, how am I going to grade those? I've been trying to hammer a good technique into you every time you do one. So if you've got poor technique, I might take some points off. I don't typically take points off for not getting isolated colonies, just because some organisms isolate way easier than others. So I can tell, where your streaks are, and we've got some more time to practice before you actually have to do it. So don't be afraid to ask me, hey, is this a good streak plate? I'd be glad to tell you what I think about that. But because we are only setting aside 3 lab periods for unknowns, you really are not going to have time to do do-overs just to show me a good streak plate. So try to do a good one the first time. Typically, if you do misidentify your unknown, I usually take off around 8 points, but that can be a little bit variable depending on kind of what went wrong. Okay, so if there's something that, this test wasn't working for a large number of people, maybe it would be less than that. Okay. And then in general, are you following lab rules? Are you disposing of your tubes and plates correctly? Are you wiping the writing off? Are you wearing your safety goggles? I don't think this open-toed shoes is going to be an issue. I think it's pretty cold. We don't have to worry about that. I typically take off points for superfluous tests. Superfluous, I'll pronounce that right. What does that mean? Well, this semester, that basically means don't inoculate things in duplicate. Sometimes students get in their mind, well, I'm just going to do 2 McConkey plates, and then if I'm not sure whether I did one right, I'll have the other one to look at. Okay, we're not going to do that. We're not going to do things in duplicate because that would waste a lot of media. I do let you go back and repeat something if you are pretty sure maybe you did something wrong. Oh, I forgot to keep the cap loose, or whatever you think you might have done wrong. You can do a test over again if you absolutely need to. We don't like students to just inoculate a whole slew of tests at once without having any kind of direction as to what they're going to do. Usually we like to have you inoculate a test, read the results, and then decide what you need to do next. Again, we're kind of compressed this semester, so I'm going to be a little bit more lenient about that. For instance, if you know you have a Staphylococcus, go ahead and do the novobiocin test and the coagulase test on the same day, and then you'll have your result the next time. So it's all right to do kind of several steps in your flowchart, in other words. Okay, but again, don't be afraid to ask me about that. When is this all due? I would like to have it on or before Wednesday, and that is the first week that you're back after Thanksgiving. Okay, and what you'll be turning in, if you look at page 79 in your lab manual, that's basically what you need to turn in along with your flow charts. So again, your gram-positive flow chart and your gram-negative. This page, again, it's basically just a list of the tests that you did telling me the results. Urea test, positive, hot pink color change. Okay, that's an example. Up in the corner, there's a little line. It says streak plate and another line for gram stain. Some of the other instructors check off right on that page whether you've done those things or not. I typically have my own way to take notes, so I don't really worry about that. Okay. Remember that we're going to be virtual after Thanksgiving, so when you turn those in, either send those to me electronically or just take a picture of it if you hand wrote it, and send it to me. Either way works. You may be done before break and you can just hand it to me. That would be great. Okay. I do, as you've been doing all semester, anything that you inoculate, make sure you've got your initials on there, the section, as always, and the date can be helpful for me sometimes. For me, if you have several plates, then I can be sure I understand which one you did earliest and which one was later. Sometimes it's helpful to have your unknown number on there as well, just in case we have any students with the same initials, so I know who did what, because I will know your unknown number as well. I like to keep track of what you inoculate because, again, just so I can kind of understand whether you're doing crazy things that don't make any sense. Not that you would do that. All right. You don't have to have all these results memorized. You can use your notes, the lab manual, you can Google things. I would really caution you against working in groups. This really should be your own work. If somebody hasn't been doing the work or paying attention or whatever, you shouldn't be doing their work for you. You can ask me questions, but at this point, I'm not probably going to be very much help. If you think you might have some kind of contamination issue, by all means ask me and I can guide you as to what's to be done. If you didn't get any growth or something crazy happens, by all means I'll help you figure out what to do there. But to ask me, what do you think? Is this pink? I'm not going to be any help with that. You're going to have to figure that interpretation out on your own. Okay, once you read a test, say you did an indol test and you've read the results, you can throw that away. You don't need to show me what that looks like. But I would like to keep what we call your stock plates. So those 2 plates that you inoculated from your original broth tube, keep those. Occasionally, even after you turn it in, because if a student misses their unknown, I can tell a lot by looking at those 2 plates. There's been cases where my key was wrong. We made a mistake in the prep room. And I can tell by looking at those plates, oh yeah, you actually had what you said it was and not what I thought it was. So don't throw those plates away, but everything else you can throw away. Okay, I will have different choices out as to how to incubate things. Okay, so it can be in a rack. This is the temperature we always incubate things. You might think it would grow better in a candle jar. So be thinking about that. These original plates, since you're going to use those and go back to those plates every time you need to inoculate a new test, you're going to want to store those in the refrigerator. If you kept those incubating for a couple weeks, then things wouldn't be alive very quickly. So Storage goes in the refrigerator. Anything you want to grow, you're going to put in the incubator. Okay. Let's see. I talked about this a little bit already. Page 79. We don't give you a lot of room there to make a big description about your results, but try to give me more than positive or negative. Give me what color it is, or vigorous bubbling, or whatever. Okay, your final exam, I haven't talked much about this yet. I've decided to open it that entire week. That's the last week of class. And different instructors for 213 are handling this differently. Some are giving the exam in finals week, some are giving it this week, some the week before. So don't pay attention to what anybody else is doing. I am going to give you some flexibility as to when you take it. Please, if you take it early in the week, please don't share with everyone else what's on the test for later on in the week. That's certainly not going to help you to do that. So just let's try to keep the exam, keep it as protected as much as we can, all right? Since you'll be at home or anyway not in the classroom, you're going to have to do that by respondus, so make sure you're equipped to do that. Okay, thanks for listening. No quiz over this. This was just, information for you so you can do a good job on the unknown. Don't be afraid to ask any questions that you have.